Chapter 18. The Designs of Life. All known species represent diverging paths of change. Life forms create a breathtaking landscape filled with elegant, powerful, and beautiful structures, features, colors, and shapes. The diversity of life on Earth is truly fascinating and magical. Instead of referring to them as living organisms, I would use the term propagating entities. These entities can be classified as either cellular or acellular propagating entities. All known propagation in the current world depends on cells, as no entity propagates without them. A cell is a membrane-bound structure that provides an optimal microenvironment for various enzymatic reactions, such as replication, transcription, and metabolism. Acellular propagating entities also rely on cellular entities for propagation, functioning as parasites on them. For example, viroids, plasmids, and viruses propagate within their respective host cells. Therefore, contemporary acellular propagating entities may not necessarily be the primordial replicators, as primordial replicators must have propagated even before the emergence of cellular propagating entities. Since the emergence of propagating entities, complexity has increased. All propagating entities possess nucleic acid-based genetic information, and their genomes have elements where replication begins, referred to as the origin of replication. Whether cellular or acellular, all replication occurs within cells. The objective of this chapter is to progressively explore the simplest to the most complex propagating entities in terms of genetic information, structure, complexity, and abilities. Essentially, this is a recapitulation of how life may have increased in complexity over time. Regardless of composition, complexity, and conditions, entities that replicate more than they degrade will exist longer. All propagating entities will either have an enhanced replication rate or a reduced degradation rate. It is impossible to cover everything in one chapter. This chapter aims to provide ideas and essential facts for a better understanding of the subsequent chapters. It aims to comprehend the incremental complexity as life forms emerge over time, focusing on the most notable examples. It should be acknowledged that there are exceptions to the general facts presented, but this overview suffices for this book. We will concentrate on the basic design, growth, and reproduction of viroids, plasmids, viruses, protists, fungi, plants, animals, and humans. Khan Academy's video on the Introduction to Biology states, "So welcome to Khan Academy's biology section. I think you're going to find it fascinating. You are going to realize that biology in some way is the most complex of the sciences, and in a lot of ways, the one that we understand the least. It is going to be built on top of chemistry, which in turn is built on top of physics, which in turn is built on top of mathematics. Biology is the study of information transfer, change, and manifestation, as well as the study of emergence, uncanny entities, and autonomous existence. There is no end to studying biology because as we live our daily lives, the different forms of organisms are changing and new ones are forming. Unless humans eradicate other species, biology keeps developing various ways and forms of existence. Therefore, I believe biology represents the highest form of intelligence. I ought to be biased. Viroids. Viroids are the simplest propagating entities, often referred to as subviral entities, and represent the most ancient forms of life. They consist solely of circular single-stranded RNA molecules that fold upon themselves through base pairing to form three-dimensional structures. Unlike viruses, viroids do not encode any proteins. Viroids are very small, ranging from 246 to 400 bases in length. They are transmitted from one plant to another by insects or other physical agents, and through plasmodesmata, which are cytoplasmic connections between adjacent cells. Viroids rely on the replication machinery of their host cells to replicate themselves, which can also interfere with the expression of vital host cell genes and lead to plant diseases. Viroids are fascinating due to their unique molecular capabilities. They comprise a single RNA molecule that performs all essential functions, such as carrying genetic information, serving as a physical entity, and enabling replication. Viroids possess binding sites for RNA polymerase, the enzyme responsible for catalyzing their replication. However, the replication process is dependent on the host RNA polymerase, which is relatively error-prone. The mistakes made by RNA polymerase during viroid replication result in mutations, which cannot be corrected. Viroids with beneficial mutations can propagate further while others perish. The most ancient propagating entities, the primordial replicator, may have resembled viroids, consisting solely of nucleic acids. Plasmids. Traditionally, plasmids have been described as dispensable circular extrachromosomal DNA molecules found in bacterial cells. This description has diminished the individuality of plasmids and relegated them to a subsidiary role in relation to bacterial cells. Plasmids have primarily been studied in terms of their usefulness to their respective hosts. Viroids and viruses are considered pathogens, but plasmids, due to their initial description, have been perceived as if they are a dispensable part of bacteria. However, plasmids are actually propagating entities with multiple additions compared to viroids. They are molecular pathogens that live off of bacteria and may occasionally confer a self-serving benefit. Most plasmids are composed of DNA. Plasmids can exist as single-stranded or double-stranded molecules, with the latter providing both structural and informational stability. The host DNA replication machinery carries out the replication of plasmids. Plasmids typically range in size from 5 to 500 kilobyte, although exceptions exist. The smallest known plasmid is 744 BP, while the largest is about 2.5 BP. Plasmids often encode multiple proteins that directly or indirectly aid in plasmid replication, stability, or propagation. The origin of replication is an essential part of a plasmid, as it is where replication is initiated. Bacteria take up plasmids from the environment through a process known as transformation. Some plasmids, through their genes, are able to form conjugation tubes that facilitate the transfer of plasmids from one cell to another. This process, known as conjugation, is traditionally, faultily, referred to as sexual behavior in bacteria. However, it is important to note that conjugation increases the number of plasmids, but not the number of bacterial cells. It is solely the propagation of plasmids, the molecular parasites. It should not be confused with the propagation of the host cells. Any exchange of bacterial genes that occurs during conjugation results from flaws in the process and should not be considered sexual reproduction. Certain plasmids encode proteins mediating fascinating phenomena, with addiction being the most intriguing. Plasmids produce proteins that would kill the host if the host lost the plasmid. Some plasmids can integrate reversibly into the host genome, merging genetic information from the plasmid and the host. That is at least one of the proposed roles of the toxin-antitoxin genes I worked with. These genes are the genetic arms that play a role in plasmid genome conflicts. Plasmid-like entities are the most primitive propagating entities that possess the ability to survive within the host cell, manipulate the host cell machinery for their own replication, and induce the host to form transfer tubes for their movement to new host cells. In terms of informational complexity, protein-coding genes have emerged. Introducing proteins into the picture marks a paradigm shift as the diversity of structures, microenvironments, and conformational dynamics becomes theoretically infinite. With multiple proteins interacting, complex reactions and structures can emerge. The probability of interacting proteins with functions increases the likelihood of complex properties in their respective encoding propagating entities. Viruses. Viruses are entities with DNA or RNA as their genome, which can be single-stranded or double-stranded. A fundamental distinction between viruses and plasmids is the presence of proteins in viruses. Viruses contain a core containing genetic information, nucleic acid, and an outer protein coat, capsid, when outside host cells. Some viruses, known as enveloped viruses, have membranes composed of lipids. Viruses can infect all types of cells or organisms, including bacteria, bacteriophages, archaea, fungi, mycophages, algae, phycophages, animals, and plants. The size and shape of viruses can vary greatly. Viruses enter target cells through interactions between the coat protein and a specific protein on the target cell. The viral genome is then transcribed, replicated, and released. Additional viral genomes and proteins are synthesized, leading to the assembly of new viruses. Viruses act as parasites, hijacking their host cell's resources and replication machinery, ultimately leading to cell lysis. Similar to certain plasmids, viral genomes can integrate into the host genome and remain dormant for multiple generations. Some integrated viral genomes lose the ability to excise themselves and permanently become part of the host genome. Approximately 8% of human DNA is estimated to trace back to such integrated viral genomes. Viruses, plasmids, and viroids are pathogens that propagate at the expense of their host cells, exploiting their resources and replication machinery. These acellular entities are obligate parasites and propagate autonomously. The origin of these acellular propagating entities is a topic of ongoing debate. It is difficult to ascertain with certainty, but viruses have likely co-evolved with their hosts since the emergence of cellular life, given the presence of viruses for every type of organism. The genetic interaction between cellular and acellular propagating entities represents an ongoing genetic conflict. When considering the emergence of life, it is challenging to incorporate viruses into the origin of cellular life. Viruses consist of nucleic acids and proteins but lack cytoplasm or an equivalent structure. This raises questions about the origin of the proteins and how multiple copies of the same protein arose. Genetic information must have been present, so how was the information in the form of a nucleic acid sequence translated into amino acid sequences? Translation is a highly intricate process that requires a cytoplasm-like environment capable of supporting multiple reactions. The existence of translation implies the presence of a membrane-delimited protoplasm with a template. Bacteria and archaea, acquiring matter and energy and using them for survival and self-propagation, require multiple chemical reactions catalyzed by enzymes. A closed system, at least partially, is necessary to maintain constant, concentrated, and favorable conditions to enable the functioning of enzymes and drive reactions in a sequence. Two essential components are required: the cell boundary and the cytoplasm. The cell boundary is maintained by semi-permeable membranes that limit the flux of materials and waste. This is what distinguishes cells from acellular entities. Cells are membrane-bound structures filled with a fluid that remains relatively constant in physical and chemical properties. All cellular entities, from prokaryotes to humans, have double-stranded DNA as their genetic material. Prokaryotes are the most primitive known cells, lacking a nucleus, but the emergence of cellular life is by far the rarest and strangest event in the universe. Bacteria and archaea are the two major domains of prokaryotes. They share many similarities but also have some differences. Both have cell membranes and cell walls with genetic material present in the cytoplasm. Some bacteria have two circular chromosomes. Bacterial genomes typically consist of around 4,500 genes, which code for proteins. However, the number of genes and the genome size can vary significantly even within a species. This is particularly true for free-living bacteria. Since the emergence of cells, there has been active competition for nutrition among different species. Variants of the same species are referred to as strains. For example, two Escherichia coli bacteria, isolated from different individuals, will differ considerably in their DNA sequences. In fact, one individual's Escherichia coli can have multiple strains with significant genomic variations. Over time and several generations, if we culture a single Escherichia coli bacterial cell in a lab, we can observe differences in their genomes. Bacterial genomes are highly plastic, adaptable, and capable of additions, deletions, inversions, and substitutions. However, core genes are essential for the cell's survival, and shell genes may aid in survival in specific habitats or fluctuating environmental conditions. Core genes typically include those required for replication, transcription, translation, DNA repair, enzymes necessary for essential metabolism, and structural genes needed to form subcellular structures. Core genes are crucial for the bacteria's existence in their current form and resist evolution or extinction. Prokaryotes exhibit active self through competitive material and energy procurement, reproduction, and inhibition of other competitors' reproduction. The most efficient reproducers persist, while others may perish. Competition among coexisting bacteria in a habitat can be measured in four essential properties: fecundity, ability to reproduce faster; tolerance, ability to tolerate stress longer; repair fidelity, ability to resist genetic change; and the ability to form beneficial interactions. Reproduction is preceded by growth and nutrient acquisition. Once a bacterium reaches its potential size, it divides into two daughter cells. the parent cell ceases to exist, forming two new entities with identical genetic identities. Each new entity, upon growth, will then divide into two more entities. This process perpetuates in prokaryotes unless limited by available resources, space, or competition. Death may occur due to resource depletion, external forces, or the accumulation of mutations. Some bacteria can form spores during adverse conditions, which can remain dormant for extended periods. Existence, reproduction, and death are the phases of life. Mutations are natural for all entities, resulting from replication mistakes or mutagens. Prokaryotes possess multiple genes for recognizing and repairing this damage, which is vital for maintaining their abilities and properties. Prokaryotes also exhibit additional features of self-sufficiency, such as acquiring matter and energy, mobility, reproduction, and repair. These traits constitute life and are essential for existence. Prokaryotes have diverse lifestyles. Some rely on organic matter, others on inorganic matter, and some even utilize light, photosynthetic bacteria. Certain bacteria can move using structures called flagella. Cellular life forms efficiently tap into available energy, obtaining and transforming it to drive the reactions necessary for existence, growth, and reproduction. Archaea are prokaryotes that thrive in stringent conditions compared to most other organisms. They inhabit extreme environments characterized by high temperatures, high salinity, extreme pH levels, and pressures. All archaea are anaerobic and unable to use oxygen for energy generation. It is believed that archaea colonize habitats with low oxygen levels, allowing less competition for resources. Most bacteria can utilize atmospheric oxygen to extract energy from food efficiently. The earliest cellular life forms may have more similarities to archaea, as they did not require oxygen for metabolism. The earliest cellular forms may have been simple membrane-bound entities with fewer genes coding for replication, energy acquisition, and basic metabolism. Many traits may have been acquired over time, such as the apparatus to express genetic information, the cytoplasm to facilitate reactions, and other structures like the cell wall. Protists. Protists are considered to be the most basic or primitive eukaryotes. They are unicellular and do not fit into the categories of fungi, plants, or animals. Ancient protists likely served as the foundation for the evolution of fungi, plants, and animals, leading to organisms with increased complexity and a wide range of characteristics and abilities. Compared to prokaryotes, eukaryotic cells have a highly complex cellular structure. They contain multiple membrane-bound organelles, such as the nucleus, endoplasmic reticulum, Golgi complex, mitochondria, chloroplasts, and lysosomes. Each organelle has its specific function. The nucleus contains genetic material and involves processes like replication and transcription. The genetic material is safeguarded during specialized processes such as replication and transcription. Messenger RNA, mRNA, is exported from the nucleus, and translation occurs in the cytoplasm. Mitochondria serve as the powerhouses of the cell, generating ATP through the oxidative phosphorylation of pyruvic acid. Chloroplasts, found only in photosynthetic protists, play a role in trapping and utilizing solar energy. The evolution of eukaryotic cells is believed to have occurred through endosymbiosis, specifically the merging of an ancestral bacterial cell with an ancestral archaeal cell. This merger allowed for the emergence of eukaryotes with complex structures that capitalize on the best characteristics of prokaryotes, enabling fast and efficient metabolite utilization. The compartmentalization of metabolic reactions is more pronounced in eukaryotic cells. Glycolysis occurs in the cytoplasm, the Krebs cycle occurs in the mitochondria, and the electron transport chain occurs in the inner mitochondrial membrane. This separation reduces interference between different sets of reactions. Additionally, multiple mitochondria produce more energy, providing energy throughout the cell to sustain metabolism. The compartmentalization of subcellular structures also helps to protect the genetic material from any toxic substances that may enter the cell or be produced during metabolism. This protection allows unicellular eukaryotes to be the first cellular form capable of taking up and metabolizing complex particulate food within the cell. This ability to ingest and digest other organisms or their parts is the first true predation, which can only occur if the genome remains undamaged. Subcellular compartmentalization also enables the continuous production of energy, supporting larger cell sizes, complex structures, and new characteristics. Building and maintaining a eukaryotic cell comes with costs, but its complexity significantly prolongs its existence compared to a simple bacterium. While most protists reproduce through binary fission, like prokaryotes, some also form spores. However, unlike prokaryotes, some protists have diploid genomes, possessing two copies of genetic material. If there is only one copy of the genetic material, it is called haploid. Some protists undergo sexual reproduction, which involves the fusion, fertilization of two morphologically different cells called gametes. This process involves a reductive division called meiosis, which converts diploid cells into haploid cells. All eukaryotes have a part of their life cycle as diploid and another part as haploid, alternating between the two forms. Diploidy appears to be linked to sexual reproduction. A good example of a sexual life cycle can be seen in paramecium. Each paramecium has two diploid nuclei, the macronucleus, where gene expression occurs to build the entity, paramecium's body, and the micronucleus, which is dormant with minimal activity but involved in sexual reproduction. The sexual reproduction of paramecium resembles bacterial conjugation, where two entities come together and exchange DNA. However, the organism's genetic material is exchanged in paramecium rather than a plasmid, as in bacterial conjugation. Genetic differences, called mating types, exist between morphologically similar paramecia. Two different mating types participate in conjugation. Before conjugation, the macronucleus disintegrates. The micronuclei of each conjugating paramecium replicate and produce 4 micronuclei, of which 3 disintegrate. Why produce 4 and degrade 3 of them? Frivolous. The remaining micronucleus divides, resulting in a female pronucleus and a male pronucleus. The male pronuclei of the conjugates are exchanged, and the received male pronucleus fuses with the resident female pronucleus, forming a zygote nucleus. The conjugates separate, and their zygote nuclei undergo 3 mitotic divisions, resulting in 8 nuclei, 3 of which disintegrate. 1 micronucleus and 4 macronuclei remain. The micronuclei undergo mitotic division, and each paramecium divides into 2. This division occurs again, resulting in a total of 4 paramecia, each with a micronucleus and a macronucleus. Why so complicated drama of making, exchanging, and degrading genetic material? Sexual reproduction is complex throughout all forms of life. Conjugation between the same mating types, likely clones with identical genetic material, would waste time, matter, and energy. Without genetic variation, the process would be futile. The mating type indicates the lineage in that instance and location. 2 organisms of the same mating type must have shared similar mutations because they belong to the same lineage. Therefore, different mating types increase the probability that the prospective conjugates are distantly related, at least not immediate kin. Slime mold, Physarum polycephalum, is a unicellular multinucleated organism that grows by extending cytoplasmic projections to search for food in its surroundings. It is considered intelligent because it can learn and communicate about its environment. Interestingly, slime mold has approximately 720 different sex types, significantly more than the typical male and female designations found in most organisms. The process of sporulation in the slime mold Dictyostelium discoideum is truly fascinating. It begins with the fusion of 2 solitary haploid amoeba, forming a diploid zygote. This diploid zygote then undergoes meiosis and subsequent mitosis, while enclosed within a highly resistant wall. Once the cell wall ruptures, numerous haploid amoebae are released. The most intriguing aspect occurs when multiple amoebae come together in response to a CK-AMP trail to form a single multicellular entity capable of moving like a slug. Within this slug stage, there are sentinel amoebae that eliminate toxic substances. Eventually, a stalk is formed, and additional amoebae climb up this stalk to create the fruiting body. The fruiting body contains spores that will germinate under favorable conditions. These characteristics exemplify the essence of multicellularity. Multicellular organisms are composed of cells with identical genetic information that are physically connected, interact with one another, and possess at least 2 distinct cell types, a process known as differentiation. Despite sharing the same genetic makeup, These cells appear different due to gene expression or repression variations. The differentiated cells fulfill specialized functions that contribute to the survival and reproduction of the organism. This example illustrates the problem of defining an entity, the single cell or the slug state. Fungi. Fungi are diverse species of multicellular eukaryotes that also have intermittent unicellular stages. They possess several distinctive characteristics, including being saprophytic, consuming dead matter, engaging in extracellular digestion, having a cell wall made of chitin, and forming spores. Reproduction in fungi can occur asexually or sexually. They grow by expanding their mycelium, consisting of a thread-like structure network called hyphae. Hyphae elongate at the tips and branch out, allowing fungi to colonize their environment. Fungi obtain nutrients by secreting enzymes that break down organic matter, absorbing the nutrients through their hyphae. This enables fungi to thrive in various habitats, such as soil, decaying matter, and even as pathogens in living organisms. Fungi can reproduce either sexually or asexually, and sometimes both in their life cycle. Asexual reproduction occurs through the production of spores, which are dispersed and can germinate to form new individuals. Spores are often produced in specialized structures, such as sporangia or conidiophores. Sexual reproduction involves the fusion of two compatible hyphae from different mating types, forming a sexually reproductive structure known as a fruiting body. The fruiting body produces spores that are released into the environment, facilitating dispersal and the establishment of new fungal colonies. The life cycles of fungi can vary significantly depending on the species. Some fungi exhibit complex life cycles with alternating generations, while others primarily reproduce either asexually or sexually. Environmental conditions, such as the availability of nutrients, temperature, and humidity, can influence the growth and reproductive strategies of fungi. In the case of yeast, a type of fungus, sexual reproduction occurs through a process called mating or conjugation. Yeast cells can exist in two different mating types, A and A. During sexual reproduction, two yeast cells of opposite mating types come into contact and form a mating bridge between them. Through this bridge, genetic material is exchanged in the form of small circular DNA molecules called plasmids. The exchanged genetic material allows for recombination and genetic diversity. After the mating process, the cells separate, and each can go on to reproduce asexually by budding, producing new yeast cells with a combination of genetic traits from both mating types. Contemporary fungal organisms offer intriguing insights into the vast and diverse kingdom of fungi. One outstanding example is Armillaria ostoyi, also known as the humongous fungus, which showcases remarkable fungal longevity and expansive growth. This collective entity, located in Oregon's Malheur National Forest, covers approximately 2,000 acres and is estimated to be over 2,400 years old. Belonging to the genus Armillaria, it primarily exists as an extensive underground network of mycelial threads, occasionally giving rise to above-ground mushrooms. This ancient and colossal fungal organism demonstrates the enduring nature of fungi as it thrives and persists for millennia through interconnected mycelial networks, often in a cryptic, subterranean manner. Plants, the robust design of life. Plants, as autotrophic organisms, play a fundamental role in terrestrial ecosystems. They use solar energy, atmospheric carbon dioxide, and water to fuel their metabolic processes through photosynthesis. Unlike animals, plants face a relatively less challenging environment, as sunlight and carbon dioxide are readily available. Additionally, their ability to absorb water from the ground and essential nutrients enables them to thrive in diverse habitats. Plants are the most efficient designs of life in trapping the sunlight and ultimately the basis for most other multicellular organisms, including fungi, plants, and animals. The unique cellular structure of plants shows key features such as chloroplasts for photosynthesis and cell walls composed of cellulose. Plant growth primarily occurs at the periphery, facilitated by tissues like apical meristem and cambium. Apical meristem drives growth from the tips, while cambium widens stems and branches, forming new branches, leaves, and flowers. The vascular system, which includes the xylem and phloem, ensures the efficient transport of water and nutrients throughout the plant. Furthermore, lignified cell walls provide structural support, enabling plants to withstand environmental forces such as wind. Plant diversity is evident in their metabolism, height, life cycle, and habitats, with various forms adapted to specific environmental conditions. Some plants, such as parasites, thrive by extracting water and nutrients from host plants, showcasing their adaptability. Plants germinate from seeds, utilizing roots to absorb water and position shoots for optimal sunlight absorption. Trees, with their sturdy trunks and branches, exemplify the importance of lignified cell walls in maintaining structural integrity. Plants have diverse reproductive strategies that involve seed germination, aerial shoots, and roots. They also exhibit regenerative potential, capable of regrowing from broken branches or cut trunks. In addition to sexual reproduction through seeds, plants engage in asexual reproduction, with vegetative propagation seen in examples like bryophyllum. Plants have remarkable adaptations, such as shedding leaves in winter or reproducing through various body parts. The resilience and adaptability of some plants, which allow them to withstand extreme cold temperatures or live for thousands of years in specific habitats, highlight their importance in maintaining the delicate balance of terrestrial ecosystems. Plants primarily grow from the outside to develop foliage and maximize sunlight absorption. Two types of tissues, the apical meristem and cambium, play essential roles in growth. The apical meristem is responsible for growth from the tips, while the cambium is responsible for widening stems and branches. These tissues continually produce new cells, forming new branches, leaves, and flowers. As the plant grows taller, the girth must also increase to support its weight, especially during strong winds. There is a wide diversity of plant structures in terms of shapes and strategies. Some plants creep while others climb. Certain plants have optimal features that adapt to their environmental conditions. The characteristic feature of all plants is their ability to grow in length from the tips, producing more leaves or buds. There is a high degree of cell division at the tips of shoots and roots. Diametric growth is present but relatively minimal. Plant cells have various types, including those involved in photosynthesis, water absorption, vascular tissue for water and food transport, xylem and phloem, propagation, flowers, fruits, vegetative structures, and protection and structural strength. Once formed, the various structures of plants remain in the same arrangement relative to the ground and stem. Parts like leaves and flowers fall off. All plants go through a growth phase before flowering, and they mature to produce seeds upon fertilization. The seeds can remain dormant for a long time and are highly resilient to adverse conditions. When these seeds germinate, they give rise to the next generation of plants. Some plants have shorter lifespans, while others can live for thousands of years. Various types of plants, mostly found on land, can be seen in every habitat, displaying appropriate structures and propagation strategies. Some plants live as parasites on other plants, accessing water and nutrients from their hosts. The regenerative potential of large trees and plants is astonishing. Even if a branch is broken or the trunk is cut, the plant can produce new branches and grow back into a complete plant. Some plants can endure freezing temperatures in the Arctic, while others shed their leaves in winter and grow them back in the spring. Certain plants can generate offspring directly from body parts other than seeds. Sexual reproduction in plants. Sexual reproduction, as defined earlier, requires mixing two different genotypes typically present in two morphologically different cells. In plants, the sexual reproduction strategy seems to progressively evolve towards seeking more divergent genotypes of the same species. Unlike animals, plants do not have designated or permanent sexual organs, like the germplasm, in their architecture. Instead, the flowers serve as the reproductive organs, forming at the tips or nodes of the shoots and eventually falling off as fruits. New flowers form seasonally, consisting of brightly colored petals that attract insects and small birds, such as hummingbirds. Flowers have two independent structures. One male, anthers produce pollen, and one female, ovary produces ovules. Fertilization occurs when the pollen from the anthers reaches the ovule, leading to the formation of seeds. There are several deviations from this typical type of sexual reproduction. In some plants, the anthers are lower in height, to allow the pollen to reach the pistil, which is necessary for fertilization to occur. This requires the assistance of pollinators, increasing the probability of cross-pollination. In other cases, the anthers mature earlier and die out before the ovules mature. Conversely, in some plants, the anthers mature later than the ovules in the same flower, again increasing the probability of cross-pollination with the help of insects from different flowers. Some plants even produce two different types of flowers on the same plant, male and female. This obligates cross-pollination from other flowers. In extreme cases of cross-pollination, there are entirely male plants and female plants, which must undergo cross-pollination or else die out. Why is sexual reproduction necessary if asexual reproduction is possible? Why are there independent structures if self-pollination is sufficient? Why do the two gametes mature at different time points? Why are there separate male and female plants in some cases? What drives the need for fertilization between gametes of diverse individuals of the same species? The typical textbook answer is to increase genetic diversity, which to me is absurd. Increasing genetic diversity is changing the identity of the self.